Welcome everybody to the 29th annual, the last lecture of the 29th winter lecture series, the Maggie and Nick DeWolf lecture series in physics, sponsored by the Aspen Center for Physics. It's actually the 54th year of the Aspen Center for Physics, and I've been here for some number of those workshops over the years, and it's always a fantastic place to come and meet your colleagues and enjoy both the physical and intellectual atmosphere of the city of Aspen. Today, we're going to have a lecture about physics applied to living systems. I should introduce myself. My name is Herbert Levine. I work at Rice University. I'm co-director of a National Science Foundation Frontier Center devoted to the idea that physics can make significant contributions to our understanding of the living world. I should say it's rather interesting to me that out of the 7 lectures in this winter series, 2 have been about physics and its interface with the living world, with biological systems. And at the National Science Foundation, similarly, out of their roughly 10 frontier centers devoted to stretching the frontiers of physics in all directions, to the large regions of astronomy, to the small reaches of the inner workings of the atom, 2 out of their 10 centers are devoted, in fact, to applying physics to the living world. So today's lecture is by Professor Lisa Manning from Syracuse, who has become an expert in this nascent field of applying physics to living materials. I've wondered how someone as young as Lisa has become an expert in the field, and when looking at her biography, I discovered the answer is that she started early. As a high school student in Kentucky, she actually was a winner of the Intel Science Fair competition, getting a very early start on an illustrious career in science. After leaving high school and going to Virginia and UC Santa Barbara, she eventually wound up with her current position on the faculty of Syracuse University, where she leads a team of individuals who are applying innovative ideas from physics to living matter. Her major contributions can be thought of to lie in the area of collective phenomena of large number of individuals. Individuals can either be dead particles like sand participating in an avalanche, where the avalanche is clearly a collective phenomenon of many millions, trillions of sand grains, to traffic jams, which is a collective phenomenon of cars, to the behavior of cells in the human body, which is a collective behavior of millions and millions of cells jostling each other for position, trying to succeed on their own, but also participating in the wondrous thing we call the human body. So today, Lisa will tell us about the human body as a living material, and thank you, and join me in welcoming her to give this lecture tonight.
All right. So first, I want to thank all of you for being here. I have to say it's so fantastic to see so many people show up for a lecture about science, and I know you have a fantastic tradition here. I've been to a few of these over the years, and I hope I live up to some of the ones I've seen. I also wanted to say thank you to all of my colleagues who are here at the conference that we're at. I've learned a lot from them this week, and I really hope I do a good job of representing my excitement about the work that they're doing. And then I'll tell you a little bit about what we're doing in my group as well. And so what you're looking here at the background of this slide is actually cells moving around in a zebrafish embryo. And so I think if you look, you heard Herb, who had such a kind introduction, say that cells are jostling around, and I think you can really see that in this picture. Okay, and so we're going to get started. And the first thing I want you to think about is that in humans and in other vertebrates, the cells move around a lot. And I'm going to say sort of correctly and incorrectly. And maybe you think already that's sort of a strange statement, because go ahead, all of you, and pinch yourself gently. Okay, you can all pinch yourself gently, and you feel like a solid, right? Except for maybe your blood and a few other things, you're a solid, and that's good because you need to do things like lift up objects, right, and support stresses. But interestingly, and I'm going to try to convince you, in a lot of important biological processes, the cells have to move around a lot more than you might expect. So let's talk about some of those. So this up here is a picture of a zebrafish embryo. And this big round thing here is the yolk. And these cells that are dividing and dividing are the zebrafish embryo cells. That's going to be the zebrafish. And these are these guys when they grow up. And you can see they move around over long distances. And you're going to see the eye form up here and the tail form down here. And it's starting to look like an embryo right about now, right? And so hopefully you saw that there were these large-scale flows of cells around this entire organism. And that's really important. And in fact, the way that they move around is programmed. So the cells that are going to become your brain end up in the head of the zebrafish, and the ones that are going to become the tail end up in the tail. And it's very important that they move correctly in order to do that. Another sort of more specific example in development is the formation of organs and where they're placed in the body. So for example, all of you, or almost all people, have a heart that's on the left side, right? Go ahead, feel your heart, that's on the left side. So how does that happen? And it turns out that in many vertebrates, in mammals, there's a little organ that is roughly circular, or in some cases hemispherical, and a fluid flow inside there turns out to go counterclockwise, and that's how the heart ends up on the correct side of the body. And so the interesting thing I want you to see from this picture, this is a picture of that organ in zebrafish, and you can see a bunch of little cells dancing around out here, and you can see the extension of what's called a notochord. And I'd like you to see that there's actually a very intricate dance of the cells that form this organ, and that's important to the formation of this organ and things ending up on the correct side. So again, cell motion is important here. There's a couple of cases of diseases where cells don't move correctly. So one example that I actually found a little bit surprising when I first learned of it is asthma. So this is a picture of your lungs, and then this is sort of a blow-up, a cartoon of what the cells that line your lungs look like. And I have some colleagues who found this really neat way to take lungs from human patients, both with asthma and without asthma, and culture them at an air-liquid interface. And so this is sort of a picture of that air-liquid interface. And then they can take a bird's eye view and look down on that culture of a layer of cells. And that's what you're looking at here in these movies. And what's interesting is these movies are different days after the cells were taken out of a patient and cultured. So this is 3 days after that happened, 6 days, and 10 days. And what I hope that you can see is that in this picture, the cells are still moving around a lot. You can sort of see them swirling around. And then at day 6, maybe those of you in the front can see a little tiny bit of motion. And by day 10, they're not moving hardly at all. And so one question is we actually, I'll talk about this in more detail later, but there's actually a difference in this behavior between patients with asthma and those without. And so when these cells are not able to sort of move around correctly, or when they sort of freeze too late, then you get, that's one of the things that's associated with asthma. Okay, and so this is a picture sort of showing that. This is the tissues of non-asthma patients, and now the colors are telling you how fast the cells are moving on each of those days. So this is day 3 for a normal patient. This is the cells are moving a lot. Day 6, not at all. Day 8, not really at all. In contrast, for asthma patients, the cells are moving a whole lot, even at day 6, and then they're still moving at day 10. Okay, so we'd like to understand that sort of incorrect motion. Okay? Another interesting thing is that cells don't move correctly in cancer. And those of you who were at the question and answer session already heard a little bit about this, right? But the idea is that, of course, if you have a cancer tumor, what's really bad is when those cancer tumors leave the original tumor, they become metastatic, and they move away and start new cancers other places. And so what I'm showing you here in bright color images, and this is from the case lab, is that this is a model for normal breast cells. The coloring is false, of course, by the way. But this is a model for normal breast cells. This is one for malignant cells, which is pretty bad. And this is one for metastatic cells, which is really bad, when things can really move around and start new tumors. And then what I'm showing you down here is what happens if you take a ball of these cells and you put them together and watch what they do. And so I'm going to show you all of these movies at the same time. And what you can see is that in the normal cells, these again are balls of cells, they sort of come together and start to fuse, but don't quite finish fusing together. So it's sort of like droplets that are arrested. Okay? In contrast, in the malignant cells, they fuse together really well. So the cells, in order for that fusion to happen, the cells are moving past one another a lot, right? And then in the metastatic cancer cells, they don't really fuse together hardly at all. Okay? And so again, I just want to bring to you the point that cells that are involved in diseases don't move the way that they should be moving, right? And so we'd like to understand that. A last example is in recovery. So many of you are probably familiar with this. If you scratch your hand or something, you get a wound, and then after a few days it closes. And it turns out that sort of one of the first steps in wound healing is the cells have to move around to close the wound. And physicists have come up with some nice models where they sort of put barriers around these skin cells and pull the barriers off in a very controlled way and watch how the cells move. And that's a picture of what you're looking at here. And again, you see this swirly motion and motion of cells. So you see there's many, many examples, even though you're a solid, of where the cells in your body move around an awful lot. So the question that I'm going to ask for the rest of this lecture is, can we explain some of this behavior by thinking of a tissue as a material that either buckles or flows? What can we get out of that, right? And the first thing you should ask me is, well, there's lots of different type of materials. What do you mean by a material? You know? And I would ask a question, well, what is a fluid? How do you tell if something's a fluid? And a characteristic example is water, right? So water, you can pour it out of a cup, right, which means it starts in one place and ends up in another, okay? And another question I want to ask you is, let's say you're at the pool, or even better, at the ocean, at the beach. Is it easier to walk or run through the ocean? Walk, right? And so what that tells you is that in liquids, the force, how hard you have to push, is higher as you move faster. So it's how fast you move in fluids that causes the force. And another important point is that it doesn't return to where it started. Fluids don't end up where they started out when you apply a force. Okay? And I'd like to sort of say, too, that if you're a fluid, another signature of being a fluid is surface tension. And so surface tension is what allows sort of bugs to crawl across water. It's what gives you droplets. And the reason it occurs is because the molecules that make up a fluid want to stick to each other more than they want to stick to other things, and therefore they round up. Okay? So that's properties of fluids. That's how you tell. Now, let's think of how you can tell something's a solid. So a good example of a solid is a rubber band, right? So you stretch it, right? It's elastic. And what happens as you stretch a rubber band, right? It gets harder and harder to stretch it the further you pull, right? So in solids, the force, how hard you have to push on it, is higher as you stretch it further. So in solids, the force is proportional to how far you stretch. In fluids, it's proportional to how fast you stretch, right? And in addition, in solids, the material does return to where it started. So if I gave you a black box, and you trusted me enough to stick your hand into that black box, and I asked you, well, what's that material? You would push on it, and you would look for these things in order to figure out if it was a fluid or a solid. Okay, what I find really cool is that the large-scale behavior that we just talked about is actually related to the small-scale structure. And in many solids and fluids, that small-scale structure is really, really small at the level of atoms or molecules. And so we're talking a microscope that looks 1 million times smaller than a yard or 1,000 times smaller than a hair. That's the scale we're looking at right now. And in a solid, the atoms line up into nice, neat crystalline rows. Whereas in a fluid, the molecules that make those up sort of jingle and jumble around, and they're not very organized at all. And it turns out that you can actually, there's some beautiful mathematical theorems that tell you how exactly that large-scale macroscopic behavior is related to the small-scale structure. Okay, but there's a whole bunch of materials that defy that easy categorization, and my favorite example is sand. Okay, so sand. Let's go back to the beach. So imagine yourself at the beach again, and you can walk on sand, right? You can't walk on water, but you can walk on sand. And what does that tell you? That tells you that sand is supporting your weight, right? Which means it's a solid. It's acting as a solid. But if you go to an area on the beach and you pick up a handful, as long as that area is sort of dry sand, it will flow through your fingers like a fluid. And so that transition between fluid and solid actually happens in a surprising number of materials. And it also happens to happen in tissues. So this is a movie over here on the left of a ball of those zebrafish cells again. So this is a ball of cells, and you're going to see it be compressed, and this movie's in real time. Okay, so you see it compressed, and it will bounce back. Not all the way, but most of the way when you let it go. Does everybody see that? It bounced back. In contrast, this movie is sped up a lot. This is on the time scale of hours. And these are, again, balls of thousands and thousands of cells. And you put them next to one another. And you see that, again, they fuse together just like liquid droplets would, for example, oil droplets in water. And you can actually make this mathematically precise. The functional form of how they go together is exactly how a fluid would. So this is showing you that on long time scales, this thing is behaving like a fluid, and on short time scales, it's behaving like a solid. And I just wanted you to think for a second, well, when is it useful for a tissue to behave like a solid or behave like a fluid? And I would say the answer is sort of straightforward. Sometimes the tissues in your body need to support stresses, when you're picking up objects. Or in development, sometimes features in your body are due to buckling, and you can only buckle if you have some elastic bit, right? So those things, it's important to be solid-like. But in the processes I talked about at the beginning of this talk, some of these large-scale flows, it's really important for the tissue to behave like a fluid in order to be able to move those cells over long distances. And so maybe it's not surprising that a lot of tissues are sort of tuned near a transition between those two types, because both can be useful in different circumstances. And then you might ask the question, well, how do I tune whether one of these tissues is behaving like a fluid or a solid? Because at the end of the day, it's made up of cells, and what the cells have to do, and what do the genes inside cells have to do to give rise to those properties, which might be useful. The other thing you're probably sitting there in the audience thinking, and this was also brought up in the question and answer session earlier, is that, but wait, cells are not like other materials. They can do so many more things than grains of sand. They're very, very complex, right? And so, for example, what are some of the things cells can do that sand can't? Well, one of the things is they can tune their properties based on external inputs. And so this is some work by Herb, who introduced me, showing that there's some signaling circuits inside of cells that help make decisions as to whether cells are going to sort of stay packed into nice sort of cobblestone-looking structures, or they're going to become metastatic, or at least change their shape and be able to move around a lot. And so there's clearly signaling pathways like this. And the interesting thing about them is they allow multiple cell types to coexist. So all this is saying is when I change concentrations of some molecules, I can get some states that look purely like cobblestones, or I can get these intermediate states, or I can get these sort of very movable states. And that all depends upon sort of the external inputs to this circuit, because the circuit allows lots of different states. Another thing that you can ask is, well, what type of external inputs change the behavior of a cell? And there's lots of neat examples of this. So this is some work. This is a picture now of a single cell. So we've zoomed in a lot compared to some of the other pictures I've shown you. And this cell is growing on a surface where half of it has no ridges, and the other half has very tiny ridges. And you can sort of see the ridges. But you're not looking at the surface at all. What you're seeing is you're seeing actin, which is a particular polymer that helps give cells their structural integrity. And you can see that on the side with no ridges, it looks very, very different from the side with ridges. And in fact, you can even see, if you look closely, that there's waves of actin that travel down these ridges. And so what this hopefully convinces you of is that cells are really changing their behavior based on their local microenvironment. They're very sensitive to that. So that's really interesting. Another thing you can do to change cell behavior is vary the stiffness of the surface. So this is a surface that goes from being very soft on one side to very stiff on the other. And this is one of those experiments where they sort of have cells and then they pull off the barriers and look at what happens. And so hopefully what you can see is they all start out in the middle, and then the ones moving this way move a lot faster than the ones moving this way. So collectively, this group of cells is sensing that this is the stiff side, and they're changing their behavior to move that way, right? And you can even do some calculations to show, to sort of track all of these cells and show that they really are preferentially moving this way in the direction of stiffness. So these type of plots show you the probability that a cell will move that way, and you can see that they stick out a lot more in the direction of the stiffer part of the surface. Okay. So they change according to stiffness. Another type of input that changes cell behavior that would never occur in, for example, a grain of sand, is that cells interact with other cells. And this thing is, I find this movie particularly exciting because maybe some of you have heard recently about the microbiome and how you have a lot of bacteria that live inside you and are important for function and also can hurt you in some circumstances, right? And so one of the interesting things is that this is a picture of a zebrafish gut during development, and those little green things are bacteria. And so as you're developing, bacteria are developing and interacting with the cells at the same time and changing the behavior of these cells, or helping actually them to develop in the right way. And so I think this is also really interesting because, of course, sand could never do this, right? Okay, and I could talk for way longer than 45 minutes about all of the other stuff that cells do, including generating an extracellular matrix. And this is sort of a substance that they can then crawl on, and they can alter the extracellular matrix around them. They can grow, they can divide, they can die, et cetera, okay? So with all of that complexity in mind, how are we ever going to get a handle on the behavior in the system? And so for the rest of this talk, I'm actually going to ignore most of those things I just said. And the reason why I'm going to do that is because I want to understand if a minimal model can explain some of the features that we see in the human body. And in other words, can we get away with it? Do I need all of that complexity to explain everything? And just to be clear, you do need that complexity to explain many things. I'm not saying it's not important, but I'm just asking how far can we go? And so for the rest of the talk, I'm going to use sort of two simple ingredients. One ingredient is that cells can exert forces and move on their own. So they can either pull on a neighboring cell or on the surface or on the extracellular matrix. And the other thing I'm going to use, and I'll talk about this in detail, is that the mechanical properties of cells give them a preferred shape. So unlike grains of sand, right, cells want to be a certain shape. Okay, so let me talk about those two ingredients for a second. So one question you might ask is, what happens when objects in general generate their own forces? Because sand does not generate its own forces. And it turns out that lots of cool stuff happens. So the first movie I want to show you is a flock of birds, I believe over Rome. And you can see, I find this movie beautiful. So you can see the birds moving around. They're not really dispersing, right? They're staying together. They form a beautiful pattern. Sort of almost looks like a droplet that's flying in the air. But it's doing something a little different, too, than you would see a droplet do, right? It's moving, it's flocking, okay? And you might say, well, birds are very, very complicated. They're an entire organism. This is very complicated behavior, right? But interestingly, if you take a model, and that's what I'm going to show you a movie of up here, where all you do is say, I'm a particle and I move in a direction, okay? And then if another particle is near me, it moves in my same direction. So you sort of look at the neighbors around you and move in the direction your neighbors are moving. That's all that's to this model, okay? And if you do that, and you look at that model over time, you see that this simple model, too, will start to form swarms and even bigger flocks. And at the end of the movie, it starts to look like this movie over here on the left of the birds. And you can even do it in a model system. So this is a colloid. So now this is a microscope image again, because colloids are the particles that sit in your milk and make your milk look white. And so you can actually look and see these happen to be light-activated colloidal particles, so they move when the light is turned on. And because, again, they're injecting energy at the local scale, they can do this sort of swarming, flocking behavior too. Okay, so this might give you some hope that interesting things happen when you just have one or two simple rules. And one of the simple rules is that objects generating their own forces gives you new types of behavior that you would never see, for example, in sand. Okay? A second ingredient is an equation for the shape of cells. And I promise this is the only equation in the entire talk. So what it means is that if I have a layer of cells, a two-dimensional layer of cells, and that actually happens a lot in development and other aspects of biology, then I can actually look down from above, again, a bird's eye view, and what that packing of cells will look like is something like this. The blue is sort of nuclei, and these are the cell edges. And if I look down like that, well, two things I could measure pretty easily in an experiment would be the area of each one of these cells and the perimeter of each one of these cells, looking down from a bird's eye view. And so then I could say, well, let's say that cells, and this isn't true for all cell types, but for the cell types I'll be talking about later, the cells don't change their volume too much. And one can show that that gives rise to a term like this for the area, which says the area sort of looks like a preferred area. So this is a preferred area that the cell wants to have, and this is its actual area. Then inside of each cell, there's this amazing, intricate interplay of polymers and motors, and we call those together the cytoskeleton. And the cytoskeleton together gives cells a structural integrity. It basically it prevents other cells from bumping too much into them. It allows them to pull on other cells. It's amazing. This term here is a very simple sort of expression to say, in general, these polymer networks inside of cells, they're generally contractile, okay? And if they happen to form a ring, which they tend to do in certain cell types, then this looks like this. And finally, cells in a dense layer like this have adhesive molecules on their edges. And so these adhesive molecules make the cells want to stick together, right? Stick. And it turns out that when you have cells that want to stick together, they want to increase their perimeter in contact. And so that's competing, the fact that cells want to stick together is competing with the integrity of the cell, the cytoskeleton. And so this term here is actually an interplay between the adhesion and that cytoskeletal activity. Okay? And so once you have written down an equation like this, which is sort of an energy for a cell shape, it has this model has one really important parameter that I'm going to talk about a little bit more. So I want to point it out to you now. It's called, I'll call it the target shape index. And what it is it's basically the target perimeter. So this is the perimeter that the cell wants to have due to all of those adhesion and cytoskeletal forces. And then a target area, which has something to do with those, and also the volume of the cell. And to make a dimensionless number, we basically want to say, okay, we have a perimeter divided by the square root of the area. Okay, so that's a target shape index. And although this isn't always true, we tend to think of it increasing as I increase the adhesion of the cells, because the cells want to have a larger perimeter when there's more adhesion. Okay, so the two questions I'm going to ask now is, are the large-scale mechanical properties of a tissue reflected in the small-scale shape of cells? Because remember, I told you for normal materials, I can look at the microstructure and predict what the macrostructure will be. And the second question that I want to ask is, are these physical properties important for their function? Okay, and so maybe a hint, I wouldn't be talking unless the answer to those were yes. And you might already believe that already, because we showed you that cells are sensitive to the surface that they're sitting on. And so now if they're embedded in a tissue and that tissue stiffens or becomes less stiff, the cells are sensitive to that, and so that has to be important for the biological function. Okay. So I'm going to tell you 3 little stories from my research group where the behavior of the material properties of the tissue are important for a biological process. So one is, again, the zebrafish development. I already showed you that. And it turns out right now, during the stage back a little bit in the movie, let me see if I can actually bring that up again for you, sort of here. It turns out that right here, in that picture, if you zoom in and now look at the cells, so these are nuclei inside of cells, you can see that what happens is certain cells are moving downward, while other cells are sort of moving upward. And these cells up here are going to become the gut, and the cells that keep moving downward are going to become the skin and brain of the zebrafish. And so it turns out that this sort of motion of certain cells going up and certain cells going down is really important for avoiding congenital disease. Okay? And so the interesting thing is that over 50 years ago now, Mal Steinberg suggested sort of a very brave, I think, conjecture, which was that maybe the reason that these tissues move like this is just like what would happen if I have two liquids and I put them together. Because what happens if I have two liquids where one has a higher surface tension and one has a lower surface tension is the one with the higher surface tension will be in the middle, and the one on the, that has the lower surface tension will engulf it, be on the outside. And so he said, well, maybe this is a wetting process. It's like the engulfment of a tissue with a higher surface tension of one of a lower surface tension. And so then you can do an experiment like this, where the cells labeled green are these cells that are moving upward, and the cells labeled red are the cells that would move downward. And then you can sort of take them out of the embryo and dissociate them and mix them together and ask what spontaneously happens in the absence of any signaling gradients or anything else. And what happens is they spontaneously sort, so the red go to the inside and the green go to the outside. And then you can actually go and measure the surface tension of these tissues by compressing them and doing some things that physicists have come up with. And you can measure the surface tension of the red tissue, and it's always higher than the surface tension of the green tissue. And this is just two different ways of generating that red tissue. Okay, so this really tells you that, yes, in fact, this tissue surface tension may be an explanation for this. It's certainly consistent. And you can use that energy model, that equation that I told you about a few slides ago, to actually try to figure out the relationship between the small-scale structure and the large-scale property of tissue surface tension. And so don't really pay attention to any of this jazz. The interesting thing is that for aggregates that have a low surface tension, that model that I talked about for shapes of individual cells predicts that the cells on the edges of the aggregates will be bubbly, really bubbly. So hopefully you can see that here. And this is a high surface tension aggregate. And then this means that the model predicts that this will be very smooth surface. And then you can actually go and look at aggregates that have a high and a low surface tension. And this is some images of those. And you can see that the model really does a good job of predicting sort of what's going to happen to the microstructure based for that macroscopic property. So that's kind of interesting. Another thing that you can do is you can try to understand this problem, this asthma problem that I talked about. So this is a second sort of vignette. So this is, again, non-asthma patients, and this is asthma patients. And again, these are different days that these things have been cultured outside of a patient. And so just to remind you, on day 6 for a normal patient, the cells aren't moving at all. Whereas for a day 6 of an asthma patient, the cells are still moving a lot. And so the question is, can we explain that difference in behavior? The other thing I want to point out is that this thing looks like it's undergoing a fluid to solid transition in real time, right? The cells are moving a lot here, and then finally they're not moving very much here. And similarly for the non-asthma patients. And so we actually can use tools from physics to precisely quantify, by looking at the motion of cells very carefully and tracking their trajectories, when this tissue goes from being a fluid to solid. And there's a precise transition. So I'm not going to show you any of that data, but the bottom line is this thing is a solid, and this thing is almost a solid, but not quite. It's still a fluid. And we can show that very precisely. And similarly, this thing is a fluid, and this thing is actually still not quite a solid, but almost. Okay? So we'd like to try to understand that using this model that I told you about again, this energy for a cell shape model. And this is a picture of a single cell moving through a tissue. At least that's what it looks like, right? But actually, that's a trick. This is just an animation, and it's an animation of the following thing in a mathematical model. So what it is it says, OK, I think that in order for a cell to move in one of these sort of layers of tissue, what it has to do is it has to change its neighbors. And it turns out it can only do that by what's called a T1 transition. And a T1 transition is when two cells start out touching, and then the edge between them shrinks, and then two other cells touch, and the ones that were originally touching are no longer touching. And all this movie is here at the bottom, let's see if I can get it to play again, is a series of those T1 transitions. So it's showing you that I just have to do these things in order, this move over and over again to get a cell to move through a tissue. Okay? And the reason that's interesting is because we can precisely calculate, using that mathematical model I showed you, how much work is required to do this. And then we can ask, well, if it costs a lot of work for a cell to move through a tissue, then that thing is a solid. And if it only costs a little work for a cell to move through a tissue, then that thing is a fluid. And we can actually precisely quantify that, and we've done that. And the thing I want to share with you today is we also sort of didn't realize it, but this model gives you a really interesting prediction for experiments. And this is the prediction based on all of that I just showed you, which is that P0 is this important model parameter I told you about. It's the perimeter that a cell wants to have based on all the cytoskeleton and adhesion, divided by the square root of the area a cell wants to have. So that's sort of what the cell wants. But you can go into an experiment or a simulation, and you can measure the actual perimeter and area of a cell, right? I showed you that's something that you could do pretty easily, okay? And then we're going to look at lots of those, because we can take pictures of lots of cells. And so that's going to be this P here. It's sort of, if I have a P without the 0 on it, that's what the cells actually are, as opposed to what they want. And then you can ask the question, does this change as you go from a fluid to a solid? And the answer is yes. And so this plot looks really complicated, but all it's saying is this P0, this is what the cell wants, and this is what the cell is, okay? And in the fluid-like region, in our model, this P, what the cell is, matches what the cell wants, because it goes up as a straight line. So the cell is getting what it wants in the fluid. But in the solid, it doesn't get what it wants. In fact, it's stuck at a number, and that number happens to be 3.81. Okay? And that doesn't depend on any model parameters. I told you there was model parameters. This is universal. It doesn't depend on anything. So it's sort of a prediction that's either right or wrong, because there's nothing you can mess with. Okay? And so a jammed tissue, a solid tissue, should have this value that's precisely 3.81. And as the tissue becomes unjammed, this should increase above 3.81. Okay, so we have these pictures of asthma tissues. We can do this. Okay, so we do it. Just as a reminder, I'm not going to show you the data for this, but this thing is solid. We have sort of proof from the cell trajectories that this thing is a solid. This thing is almost solid. This thing is almost solid. And then these things over here are fluid. Okay, so what the model predicts is that if I'm a solid, if I'm jammed, I should sit here precisely at 3.81. And as I become unjammed, I should move away from that. Right? And similarly here, I should be close to here and then move away from that. And this is what the data looks like from the asthma tissue. And I have to say, when I saw this at first, I didn't believe it, because it's hard to predict something in biology. And by the way, this shouldn't work for every tissue. It happens, this asthma tissue happens to have certain properties that make this model work really well for it, I think. But in any case, it sort of moves away, and it's really this number when it's jammed, which I think is pretty neat. And so then you can start to ask yourself, well, now that I understand sort of better that this jamming liquid to solid transition is very different between patients with asthma and those without asthma, I can do a few things. I can think, well, what sort of drugs will affect this cell shape perimeter, right, parameter? And also I can say, I can use this to sort of see whether a treatment like a steroid is working well or poorly, because now instead of having to take lots and lots of movies of cells, which takes a lot of time, I can actually just look at cell shapes, which I can just look at a picture. So this tells you you can look at a picture of a tissue and figure out what its mechanical properties are, at least in some cases. Okay, so the last, with physics. So the last vignette I want to tell you about is about cancer. And this is some work with Joseph Case's group and other collaborators. The question that we're going to try to ask is, can we find signatures for cancer invasiveness? And by the way, that's the answer to this right now is no. This is something we're going after, and I just wanted to tell you how we're going after it. Right? And so the first thing I want to tell you that why I'm super excited to be working on this is we're working with primary tumor cells from patients with breast cancer and cervical cancer. And the reason it's important to work with primary cells is because, as I showed you, the cells change their behavior depending on their environment. So if you take cells out of a patient and then you culture it a lot and culture it again and culture it again, those cells are living in like some weirdo water environment instead of the environment they were in before, and they change their properties, right? So it's important, if you're going to do this sort of thing, to work on primary tumors, although we don't always, because cell lines are useful for things sometimes. Okay, So the idea is you can take one of these primary tumor cells, you can dissociate it, and then you can send it through a laser beam. And all the laser beam does is stretch the cells, and it looks, and you, under a microscope, look at how far the cells stretch. And that's going to tell you how stiff or soft an individual cell is. And also, this is fast, so you can test lots and lots of cells in a primary tumor. And so you get a bunch of curves that look like this. On this side is the deformation. And so what you're looking at is if a cell, one of these things deforms a little bit, right? That means that the cell is stiff, because you're applying the same force and it only deforms a little bit, okay? In these dark blue curves, if I apply the same force, the cell stretches a lot. So that's a softer cell. So this is the way we tell stiff and soft cells, and we can get a whole population of how stiff or how soft are cells in these tumors. And what you find generically is that in the cancer tissues, there's a lot more softer cells. So again, this is how much deformation there is. So more deformation means softer. Okay, so the dark blue, which is cancer, has a different distribution, and it's a lot softer than the the controls in both breast cancer and cervical cancer. Okay, and so we'd like to ask, you know, if we have all of these soft cells, why is the tumor really very rigid? And the first thing I should tell you is that most of the reason a cancer tumor is rigid is because of the extracellular matrix that surrounds these tissues and because of the way they're pulling on it. But another question you might ask is, well, if I have a solid mass and it's made up of a lot of soft cells, can it still be rigid? Can it still support stresses? That's kind of weird, right? And so we looked into that, and this plot is a little bit scary, but what it means is this axis is how stiff the tumor is. Okay, and again, this is in the model, within the framework of that energy model that I told you about, how stiff the tumor is, and this is the fraction of stiff cells. And so a cell is stiff if it sort of has this perimeter to area ratio that's sort of less than 3.81, and soft otherwise. That's how we're characterizing them. And sort of, this was very surprising to me. What this plot tells you, because it sort of comes down a little bit before 0.5, which is 50%, it tells you that a tumor can be rigid, it can be stiff, even when more than 50% of its cells are soft. That's kind of neat. And the reason that happens is because there's actually a percolating network of stiff cells in the tissue that supports that rigid stress. And so that's neat. But it also makes you think there's a pattern of rigid cells now inside this tumor, if you believe this sort of modeling. And then you can ask, well, what does that mean for the soft cells that are left over that aren't part of this rigid network, right? And so what happens, this is a movie, so what happens is if I have some soft cells, so in blue are softer cells, and the surrounding around it is stiffer cells. I'm not drawing those in for you, but there's a lot of cells that aren't colored in here. And what I think you might be able to see from the beginning of this movie is that there starts to be a pack of soft cells at the beginning. Let's see if I can make it play again. And then there's streams of soft cells that sort of come out of that tumor. And why that's interesting is it seems that these soft cells are finding these sort of one-dimensional string-like pathways out of the tumor that are sort of generated by this rigid backbone, right? They find these sort of one-dimensional strings. And the reason that sort of, and this plot is just quantifying that effect. We can say that somewhat quantitatively. And that's interesting because that sort of thing where sort of there's streams of cells that are exiting a tumor, it's called multicellular streaming, has been seen in some cancer tumors. And so the idea is that the heterogeneity in mechanical properties of cancer, and that's sort of well known, that cancer tissues are sort of, they're heterogeneous biochemically and mechanically, that heterogeneity actually sets up a perfect storm. It allows the tissue to sort of remain rigid so it can pull on the extracellular matrix, but it allows these streams of soft cells, which are the bad ones, to sort of get right out and go in streams and go elsewhere. So this is sort of interesting and suggestive. It's not a full story yet, but it's something we're thinking about. Okay, so in summary, the tissues in your body can, I hope I've convinced you, that it's interesting to think about them as being a living material. And sometimes it's useful for these tissues to be fluid-like, and sometimes it's useful for them to be solid-like. That's sort of important for their biological function. That we can use simple models from sort of materials physics to make actually incredibly accurate predictions about the behaviors of these tissues. We can hopefully use some of these insights to understand disease, and there's a lot more exciting things to do. So I first want to say thank you to all of the people who helped me do this work. Maybe I'm a theoretical physicist, and you might think of a theoretical physicist as somebody that works like by themselves in a little room, and that's not true at all. So I have all of these people to thank, and I can't fit their pictures. In particular, I want to thank my excellent former postdoc, Max B., who's here in the audience, who did a lot of this work, and other folks in the audience too. And the last thing I wanted to leave you with is thank you for your attention. And I also wanted to point out that all of this work, or a large part of it, has been funded actually by the federal government. So I want to thank you too for your taxes, because this type of work is only possible when I think the federal government supports it. So I want to thank you for that too.
I guess we have time for a few questions. Before that, I just want to say that after this, there is a free reception. So down the street at 300 Hyman, Steam Room, sponsored by the Aspen Physics Cafe organization, Aspen Science Center. So please attend if you're interested in that. And I think we have time for a few questions for Lisa. So there's one over here.
So your talk begs a certain question to arise, and that is, when someone dies, in a car accident, or attack, or whatever, how do the trillions of cells that are sharing this energy and change movement know it's time to shut down and say goodbye?
Oh, so the question is what happens to all of this sort of beautiful orchestration of cell movement when you die, right? And I think one of the things is that for these cells, all of their activity is actually driven by ATP, which is a particular molecule that sort of transitions between two states. And so the idea is that if that molecule doesn't keep turning over, and the way it turns over is through all of your, you have to eat food, and then it has to be converted, that energy from the food has to be converted to changing this one molecule, ATP, ADP. And so when that process stops happening, the oxygen stops flowing, your heart stops beating, the cells don't get that molecule to turn back and forth, then all of these active processes that I told you about stop. And once they stop, because all of the cells are injecting energy at the local scale, and that's what allows these processes to happen, all of that stops.
So this formula here can actually get it down just like MC squared? I mean, it actually gets everything straight?
So the question is this equation I showed you, like E equals MC squared? And the answer is really, really no. There's a lot of people in this audience that would strongly disagree and have disagreed with me this week. And they're right. This happens to be useful as a model for certain tissues. And in fact, one might ask, and we are asking, the fact that this simple of a model works so well and predicts that number 3.81, what it really suggests, because probably our model isn't exactly right, what it really suggests is there's a deeper universality here to this liquid to solid transition in tissues. And so we can learn something from that model I wrote down, that one equation, but actually there's a deeper question of why it works so well and what sort of, how you could perturb the system, how you could change it and still end up with that answer, because clearly cells are doing stuff all the time, okay? So it's a suggestion. Right.
You have a question there? Yeah.
Well, I don't have a lot of knowledge in this subject, and I'm curious, what does it really mean to me? Is this helping cancer research? Is it helping for asthmatics? I mean, what can I take home and do with it?
Sure. So the question is what is this useful for to a normal person that's not excited about, sand? Okay, and so I think that the answer, so I won't be able to answer this as completely as maybe some even the other people that are at this conference with me, but let me tell you what I think the answer is. And I would say it is, I have a lot of colleagues that do really excellent biology experiments, but often they have trouble sort of identifying hypotheses to test to see what will work to help affect cancer. right? If I'm developing a cancer treatment. Or what will work to help asthma? Right now what people do is they throw drugs at it and see what works. It's not quite true. Actually, there's some design principles for some things. But we really don't have design principles for fixing things like asthma, cancer, congenital disease. And so what I think this does is it gives you a framework for thinking about what the important parameters are to change for a single cell to change the behavior of a tissue, which is important for these diseases. So the idea is it gives you targets to think about for drugs that you might not have thought about before, right? Because you can understand fundamentally what it is that you have to change about a single cell in order to get the whole tissue to behave differently. Does that help answer the question? Yeah.
Yes.
How does this theory of the work that you've done get applied in terms of organ donation and transplants?
Oh.
It seems like there are certain rhythms there of the original organ, and you may be looking for establishing the same kind of rhythm you're looking for in the recipient that makes it more successful.
So the question is what does this work have to say about things regarding organ transplants and sort of what makes a good organ transplant? And so I should first state that I'm fairly highly unqualified to answer that question. Let me tell you one thing, which is that I do work with people that are interested in organs on a chip and making organs sort of not quite from scratch, but kind of from scratch. In other words, if I want to have, instead of waiting for somebody to have an accident or somebody to die to have an organ, instead maybe we could grow them from a chip or maybe grow them from somebody's own cells so that they don't have rejection of that organ. And so for that, models like this, we're not there yet, but models like this help you understand what the processes are that help shape organs. That's one of the things that we're thinking about doing, like what are the forces, and also what are the biochemical signaling pathways, which I sort of washed under the rug for this talk, but are very important. So one answer is this affects, I think, organ transplants by helping our ability to sort of think about growing organs. And the other thing is that there are certainly a lot of ways in which sort of organs that you take from one person and put in another person depend on sort of, you have to have the right match between the donors and things like that. And I don't think that this work has very much to say about that aspect.
Any, yes. Yes.
What is the relationship between genes and cells in this work?
Oh, that's a really good question. So the question is, what's the relationship between genes and cells? And the first thing is that at the end of the day, you know, when you have a fertilized egg, all of the instructions for what the embryo is going to become is in the genes, right? So it's important to note that at the end of the day, all of this is controlled by genes and environment, right? You can perturb things by the environment as well. So what I would say is, the way that I think about it anyway, is that the genes control the mechanical properties of single cells. That's sort of what genes can do. And there's all of these signaling networks and proteins that are expressed. For example, the sticky molecules that sort of stick two cells together, they're called cadherins. There's a particular gene that encodes for them, for example, okay? And so if you express more of that gene, you get more of those adhesion sites and you change the cell shape, for example. And so the idea is that there's a program that's carried out that says like how these genes are read off. And then the question is how do those sort of entities, cells, self-organize to generate patterns? Because it's hard to believe that all of the patterning comes programmed from the genes in the sense that this cell goes to position X and this cell goes to position Y. Instead, what makes more sense and is more robust is that the genes sort of tell single cells what to be, and then they sort of self-organize and form patterns to generate structures that are useful for biology. Does that sort of answer your question? Yeah. Biology is complicated.
Maybe time for one or two more, but yes, go ahead.
You showed us a cell related to embryonic development, where certain cells were going in one direction and other cells were going sort of counterclockwise, if you will.
Yes.
Are those cells self-propelled, and what signals are they following?
Oh, that's a really great question. So the question is, I don't think I'll be able to make it back, so sorry. But I had that movie of the red nuclei stain, and some go down and some sort of go up the other way, right? And the question is are those particles or cells self-propelled, and what are their instructions? And the answer is they are certainly self-propelled in the sense that they are crawling past one another by sort of generating sort of adhesions and pulling on one another. We know that. And so that's what allows them to sort of exert forces on one another. In addition, it seems that once you sort of have enough self-propulsion in the system so that the cells are sort of jiggling around, it gives the tissue a sort of a fluid-like behavior. And one idea that I proposed and sort of provided some evidence for is that what actually happens is the cells are sort of all the time touching each other and sort of figuring out where's the best state for them to be, where sort of they have to do the least amount of work. Okay? And so if you sort of believe that they're sort of jiggling around and doing sort of the least amount of work they have to do, then physics tells you that these two tissues have a surface tension, and just sort of because they're jiggling around and they behave like a fluid, they'll spontaneously wet one another. So you can do an experiment at home where you sort of take oil and water or some immiscible fluids and sort of ask what happens when one of them wets the other, and it's sort of exactly what you see there. Sort of.
Okay, I guess one last question there.
Do electronic pulse influence any of that behavior?
Sorry, can you repeat?
Exterior electronic pulse influence some of that behavior?
Oh, an electric. Like, what's the effect of an electric pulse on all of this? And so the answer to that question is I am not an expert on that at all. But I do know some people who are experts on that, and I do know that there's a big change in the behavior of many cell types when there's sort of an applied electric field. But beyond that, I can't say anything more. They're very sensitive to, some cell types are sensitive to electric fields, but that's all I got.
Okay, well, let's thank Lisa again for giving the talk. Okay.